Hello, in this video we're going to look at cell junctions and then just the idea of what chapter 6 on cell communication is talking about. Cell junctions occur in animal cells where you have direct cell-to-cell contact. This would be in contrast to what we talked about in chapter 4, where we talked about cell coverings, things like the extracellular matrix, that grid work of proteins and polysaccharides located in between the cells where there's a little bit of space between the cells. Okay, as we go through this, pay particular attention to the material that's highlighted in red. Now, with the animal cells, one type of cell junction we have is what's called anchoring junctions. And with these anchoring junctions, you have very strong proteins coming off the plasma membrane, and these act like Velcro. When you smush two pieces of Velcro together, they have little hook structures that intertwine. That's the same thing these proteins do. Little hook structures are going to hook into each other, intertwine with each other, and hold these two cells together. Now, these proteins are also connected to the cytoskeleton inside the cell, and specifically it's connected to the intermediate filaments of the cytoskeleton. Remember those intermediate filaments? Those are the parts of the cytoskeleton, those fibrous proteins in the cell that they're not functioning in movement. They are functioning in maintaining the structure and the strength of the cell, and they are unique to animal cells. Okay, so we've got these strong proteins that extend to the outside of the cell and hook in with one another. They're connected into the intermediate filaments inside the cell that strengthen and reinforce the cell from the inside. And then what this does is leaves a little bit of space in between those adjacent cells, and that little bit of space makes these cells flexible, and it allows them to stretch. And so that's where this is found, in tissues that have to be flexible and able to stretch. Things like the skin and the urinary bladder. Okay, here's a diagram from your textbook showing the idea of anchoring junctions. And right in here, let's draw around that. So right in here is the idea of these proteins that come out and connect in with one another, hook together like Velcro. Okay, so let's look at another one. Notice this one happens to be in red, so you might want to pay a little more attention to this one. So in animal cells, another type of cell junction we have is what's called tight junction, and the name is pretty descriptive. This is where the plasma membranes of adjacent cells are held together with no space in between the cells. Thus, they are held together very tightly, and it's tight enough that it produces waterproof junctions. And this will force materials to pass through the cells rather than being able to pass in between the cells. Now, one place we find this is in the cells lining our intestines. So they're in the small intestines. You don't want that fluid from the small intestines making its way in between the cells and into your body. You want that material to actually have to go through the cells so that those cells can have a little bit of selectivity as to what's going to get from the digestive tract out into your bloodstream. Now, here's the example from your textbook that shows this, and up here you see an example of the epithelial cells lining the small intestines. You got the little microvilli there, and then here's an electron micrograph showing the same thing. And right in here and right in here, you see that area where you have tight junction, where those plasma membranes of the adjacent cells are actually stitched together. And this is the way it's going to work. You're going to have these proteins extending between the two cells that stitch them together very tightly and make a watertight barrier. Okay, the other type of junction that we want to look at in animal cells is what's called gap junctions. These gap junctions are very tiny pores between adjacent animal cells. And these pores allow for the very rapid movement of small molecules, in other words, chemical substances, and ions. So things that have an electrical charge to them. And it's going to allow these to move from one cell to another, and that's going to serve as a means of cell-to-cell communication. Now, where you're going to find this is going to be in things like cardiac muscle, where you want this tissue to act in a very coordinated manner. Okay, so here's a diagram showing the idea of these gap junctions. So right here, you can probably see it best in the artist version of this, where you have these proteins in the two cells, and those connect together, and it forms a very tiny channel between the adjacent cells. Okay. Now let's move from animal cells to the plant cells. Notice again, this is in red. Who knows, you might see this again, maybe on Friday. So dealing with the plant cells, we have what's called plasmodesmata. And this is sort of the plant version of the gap junction. And so here you have tiny pores that are going to go through the cell walls between adjacent plant cells. And what this does, it connects the cytoplasm of one cell into the cytoplasm of the next cell, and it allows small molecules to move from one cell to the next. Again, it acts as a means of communication between cells. And so in a plant, if you just had solid walls all the way around every one of these cells, then each one of those cells would just be a little island in there to itself. And in a multicellular organism, cells do not work as little islands by themselves. Cells work in conjunction with other cells. Even in something like a plant that doesn't have a nervous system, doesn't have a brain, it's still a multicellular organism, and you have to have cell-to-cell communication. That is accomplished by, one way it's accomplished is by means of these plasmodesmata. Okay, here's a diagram from your textbook showing the idea of right here we have the cell wall of the two cells, and then right here is an opening through that cell wall. Here's another opening through that cell wall. And notice one of the things that happens is this plasma membrane of one cell comes around here, and it curves through that plasmodesmata, and it becomes plasma membrane in the other cell. Okay, same thing right here. You see plasma membrane of one cell comes around, becomes plasma membrane in the other cell. So we've got cell-to-cell communication in these, or connection, which leads to communication in these plant cells. Okay, let me turn off the drawing tool, and let's take a quick look at Chapter 6. Basically, on the test, you're going to have one question that deals with Chapter 6. It's the idea, what is the topic in Chapter 6? What does Chapter 6 deal with? And right here, Chapter 6 is dealing with cell-to-cell communication. Okay, so we have this term cell signaling. And cell signaling is a means of cell-to-cell communication, and it is essential in any multicellular organism, even those like a jellyfish or a plant that does not have a nervous system or any kind of a brain. So cells do not work in isolation, they work in conjunction with other cells. And the way cells are going to communicate is by chemical means. So a chemical substance is released from one cell. This chemical substance will then attach to receptor proteins. Notice that's in red. So these receptor proteins in the second cell will pick up the chemical substance released from the first cell, and this chemical substance acts as a ligand. Remember, a ligand is a term for anything that is picked up by these receptor proteins. So this ligand is our first messenger. Then the receptor combined with the ligand will stimulate what's called signal transduction. Now, signal transduction, this is where a new chemical signal, something different from the ligand that's held in the receptor protein, This new chemical signal is released inside the cell. This new chemical signal that is now released within the cell is now our second messenger. Okay? And we don't need to worry about this part down here. Let's just go on. Okay, the second messenger is going to activate a process within the cell. For example, it might activate some kind of chemical activity in the cell that would change the what's going on in the cell, or it might activate the reading of a certain part of the DNA, which would in turn change the activities of the cell. Okay, well, that's going to wrap us up. Good luck on the test on Friday.